All right.
They're different cookies, aren't they?
Different cookies.
Yes. Right.
Wait, Ivy mixed those together.
Ah. Mix them together? Mix them up?
Literally just started. She's literally started off.
Woo!
Yeah, I have to come up with someone else.
How long is your presentation?
Probably an hour,
Want to see my name? It's written.
roughly.
Oh yeah. Yeah, whoever that is. Some person.
Okay. Yeah, I can see. Oh, no, I might just stay here.
It's on the back of her shirt. Yeah, what's that?
Yeah.
Zebra long range?
Oh yeah, it's a zebra long.
You're trying to find what that looks
Okay. Yeah, that can work. Yeah. Oh,
I want those in
let me not take over the mouse from you.
Yeah, versus fish or butterflies. Pick one.
Yeah.
So those are butterfly fish.
Butterfly fish.
Stop it. Get out.
I'm allowed to thirst.
Yeah, that's right.
No, see, it's my home state. I was there first. Ooh, when I was born there. Ooh, but I was there first. How long has your family been there? Ooh, not since the 1800s.
Wow.
So, if you were to go this route, I'm the weirdo.
This is something I just came across in my search, so I thought it would be kind of fun to share it around. This is an actual 3D model of a scanned newt skull.
And a good thing they're not much bigger.
Yeah.
Or we'd all be in trouble.
I know. I just thought this was kind of cool.
And they're pretty slow.
I wanted to show it off.
Oh, well.
Yeah.
See the little bridging there. That's not really good. That was just
Oh my goodness, that was sad.
You could play the
No, I can't.
play the original video in the loop while it's going. Oh no!
My favorite diamond is, I haven't done the whale this year, but California, they have these giant strawberry trees, they're like this long. And I can't hear you. I'll show you something. I have a whole science fair video in California. Can you use rookery?
That was rough.
Like, what?
Well, now you're playing it.
yeah, that's.
Is it hard? That project really hard.
That project could take a lot of time.
Andy on.
No. My phone is busy.
But we have in the front.
Yeah, well, I've got it streaming on there. This is recording. Yeah, I sent one to the PowerPoint. He might be on the stream system because
I'm watching a video of y'all.
oh, okay, good.
Can you hear me?
Do you need a phone?
I don't know. Is there a way that
You can see who's like logged on in with it. If he's
Oh, really?
somebody called in?
if you pull up that thing,
Yeah, okay.
It's
That's it, that's it.
Yeah, that's it.
Different types.
Okay, there's different sizes.
Testing, hear me?
Yay, right?
Yes, and what you should, here's I know.
Hello? Andy, say something.
I heard something.
Heard. Well, we should be. Yeah, well, his shirt actually has newts on it. Yeah. Take a picture of it. Or walk up to the camera with it so you can see all that. Yeah, part of showing the little threads. Yeah, I don't know. I guess we'll have to use my speakerphone. It'll be up there, well, I don't know if you can pop up a text message in there. Maybe that's what you could do to communicate, and then we'll switch over to phone once we ask questions. There's text me. You just want to text me when you have it. Well, I guess by the time we're finished, then you'll have a question, then we'll call you back. Yeah, that's what I'm doing. Yeah. Okay, we'll do that then. All right. We'll talk to you soon. Yeah. Thanks. This is the second one today. Nice. All right.
But that's not how they do it. They're supposed to do it awake or kiss.
I know. Well, this is two females, so they don't know anything. Oh, yeah.
Did you send the info again?
I guess I did. I was busy making this thing.
Just 5 or 6 pages.
Yeah, you guys are 3 chapters, though, right? For now. Okay. Yeah. Oh, yeah. Yeah. We were trying to figure out the right order to put them in because they don't really tie together so neatly.
No, well, tell Andy about the chapter 1.
Did he get it?
Well, he said he never got it either.
Wait, what? He never got chapter 1?
Well, not the intro.
Oh. Okay.
It's edited. You still say it.
Yeah. I thought I.
The only thing that might change is progression. I think the way he's presenting about makes it best. Since
Yeah.
they're almost independent things.
Doing the mapping first, followed by the genetics and the survey.
So let's see. Committee.
No, I'm also making those.
He sent me an email about the nature serve, so I added that as well. Just in brief.
It's on my list.
And yeah.
Yeah, I think
Chapter 1 is generalized.
And he was there for like 10 minutes.
It's been a challenge. It's been a busy 3 weeks.
Well, where the microphone is, I guess it's on that. I don't think those are working.
Oh, those aren't picking them
I
up?
think it's there.
Oh, okay.
Yeah,
Those are
why
working.
not, Jason?
This is one of your newts. She was there for quite a while.
We're gonna be toward the end, and then once I do that, I'll resubmit documents. They'll sign off and say, yes, this is what I'm looking for you to change. That's sort of what's get a few extra letters by
No, it's not worth it. It's not worth taking a chance.
Faster than matching.
I guess we're all here. You like the lighting?
Yeah. Mine's not a darker presentation, so. There it
Is
is.
that, bro?
There's the area. Do you have explosive new today? Fresh anything?
I'm sorry, but then I can't hear you in the back.
Too bad.
Right, you have 40.
That's new dig. It was just late. That's what I was getting at.
Can't have that going on in the background while I'm talking.
She just it's fine.
All right, welcome everyone. We're here for part 2 of the Climate Lab Symposium. Evan is now going to be defending his thesis work regarding the black-spotted newt. And Evan came to us from the University of California, Davis, where he had done some amphibian work there as well, an undergraduate. He's worked on several different species over time. Did some frog research at UC Davis with the Rio Cascade frog. Was a field assistant doing rattlesnake behavior. Did some mammal ecology as well. Working on rodent data with flower counts and species IDs. And here, Evan has done quite a bit of work on this project funded by Texas Parks and Wildlife, state wildlife grant. So this project has to do with the black-spotted newt, a state threatened species that we know very little about. Evan has made great strides in learning more about this particular species and about its distribution. While he's been here, he's presented 6 times on his research in various degrees. He's gotten several scholarships to work with the black-spotted newt, one to fund borescope work and things like that, and to attend conferences. And also, he now has 3 publications regarding some of his work that he's done here. One, a magazine article, popular article co-authored with me about the black-spotted newt and its behaviors. Also a description of its habitat in Herpetological Review, and also another publication with Clint, who's in the back here, and Max Pons, who's also back here on a lizard that they expanded the distribution. So he's done quite a bit since he's been here. But what he's going to present on today is surveying genetics and distribution of black-spotted newts. And without further ado, take it away. Well, I would like to add that one last thing I forgot. Andy Glusenkamp is on video link. He was previously the state herpetologist for Texas. He's now working in conservation at the San Antonio Zoo. He's on the committee, and also Dr. Diego Figueroa, who's also one of the committee members, and we will be judging his work for acceptance of his thesis for his degree.
So hard to judge. All right, yeah, so I'm here to talk about my work with the black-spotted newt. As I'm sure many of you are well aware, that's my main focus. So I'm going to start off with a very brief introduction so we can actually get into everything else that took place with this study. So, real quickly, black-spotted newt, it's a small to medium-sized salamander native to southern Texas and sort of North Mexico along the Gulf Coast here. It's considered to have two different subspecies, two distinct subspecies, one that is northern and one that is southern. The Mexico subspecies, photoed here by Jaime Pena, you can see it's a much darker individual than the northern subspecies, meridionalis. It's got more of a yellowish ventral side, and aside from that, like a few other morphometrics that I'm not really going to go into because that's not what this project is about. But as Dr. Klein was mentioning, this species is very poorly understood. So what I have here are the citation listings from Meacham, 1968. He was actually like the primary researcher regarding this species for many years, and this is all that he was able to find regarding black-spotted newts in 1968. But something I want to highlight, only these references actually give anything more than location data. All of the rest of these are like checklists or annotated guides on where to find things. This is all of the data that Meacham had to work off of. And the problem is, it didn't get much better. Rapol and Klicka in 1991 wrote a report for Texas State, wherein their last paragraph, further problem regarding the newt is that it is practically unknown. Examination of the attached bibliography has less than 10 papers that deal specifically with this newt, and these relate mainly to taxonomy and distribution. They actually go on to say that much of what is understood about this species is actually brought over from what is known of the eastern newt, the sister species. Now, for my study, I wanted to do a more updated citation list, and these are all the papers that I was able to come up with that specifically referenced meridionalis as a species. However, even in this list, these 6 are misidentifications. So Strecker identifies individuals up in San Antonio, identifies them in, and one individual in Dallas, and then Harwood actually tried to identify some parasites, but he used individuals from Houston, and all of those are from Notothalamus viridescens louisianensis, the sister species.
Yeah, I guess that remote you're doing is moving the
Yeah.
mouse all over.
If I do it like this, it's a little better, I guess. I'll just click it when I need to. So, because of this really dearth of information, the black-spotted newt is considered very endangered. In Texas, posted here, Rule 65.175, listing it specifically as a threatened species in the state of Texas. And you can see, of those amphibians, there are about 10 listed. Black-spotted newt is on there, and actually the bottom 6 just so happen to also be natives to the lower Rio Grande Valley, where the black-spotted newt is also known, is predominantly known to be in. In Mexico, it's listed as endangered or en peligro de extinción, and that was defined in 2001, and an updated list still listed as endangered from 2010. NatureServe has them listed as critically imperiled at G1 status, and IUCN also has them listed as endangered in the wild. So we're working with relatively unknown. Rapol and Klicka, they found 7 sites. Half of those were in Mexico. And another professor, Dr. Jude, went about and wrote a report in 1985 where he found 3. He only managed to find 3 populations, 2 in Texas and 1 in Mexico. So very little is understood, and it's just difficult to find them in general. So I'm going to start off with trying to present a more modern distribution for this species. Meacham, 1968, he defined the first real range for them. He defined the first real range for them in Texas and Mexico, the map I showed earlier. But for this, we're using more modern algorithms. So I specifically use the maximum entropy algorithm, known as MaxEnt, because it is widely known to be able to work in using very few records. There are some studies that use only 5 records to define an area for where they can find the specific species. But working further with that, also tried to identify their specific like environmental characteristics. That's what this slide is for. I accidentally skipped over the first slide. But using this, try to identify like how do they relate, or how do the 2 subspecies relate through their environmental factors. Using Permanova, try to differentiate which factors are actually really driving that separation, and BioEnv as more of a supplement to Permanova analysis. So, as an initial taxon comparison, I gathered up museum records, literature records, wherever I could find them basically. And any that were cross-referenced between records and museum, that was great. Problem is, some of them don't come out that well. I'll get into that a little bit later. But working off all of that and environmental data supplied by BioClimb, the soils grid, 250 meters for soil data, and then using elevation data provided by Earth Explorer, extracted all of those values to each individual location and ran an MDS plot. And based off of this MDS plot, you can see that Viridescens louisianensis, the completely different species, very well separated from Notophthalmus meridionalis here. Now, between the 2 subspecies, you can also see that there is a pretty well-defined separation. There's a little bit of overlap here, and that's likely due to their overlap range down in Mexico. Using this, extracting the eigenvectors from that and identifying which ones had the most impact on this analysis with Permanova, the 4 that really stood out were Bio 1, Bio 4, Bio 11, and Bio 18. Annual mean temperature, temperature seasonality, mean temperature of the coldest quarter, and then precipitation of the warmest quarter. And so going a little bit farther into that, working with that same MDS plot, I overlaid the contour levels for each of those 4 variables to really drive home how these variables are different across these 3 subspecies. And so you can see we've got mean annual temperature, temperature seasonality, coldest quarter, and warmest quarter precipitation. Now, up here in the mean annual temperature, Louisianensis really has a pretty restricted range. It seems to prefer or resides in cooler temperature areas, so really between 18 and 21 degrees Celsius. Now, previous studies have found Notophthalmus meridionalis cannot handle below 10 Celsius. And really, according to this, we also see that their mean annual temperature resides much higher in like 22 degrees Celsius. Compared to, and that's, Calerti seems to sort of have this long swath that it's able to reside in, as opposed to the northern meridionalis. Temperature seasonality, again, Louisianensis is pretty distinct on its own. It shows a preference for a much higher seasonality. So, this means that winter is very different as opposed to summer. But when we get into meridionalis has a bit more of a stretch there. Calerti Clarity is all over the place. Clarity, again, can have either really high seasonality or really low seasonality, but seems to prefer this low seasonality variation. Mean temperature of the coldest quarter, again, Louisianensis, pretty cold. It can survive harsher, it can survive much colder conditions. In fact, I've seen video of them actually swimming around under ice. Meridionalis would die in those sorts of conditions. And again, in here, we can see 14 to 19 Celsius for the coldest quarter. Calurdi seems to actually prefer these higher temperatures. Most of their individuals are right in that center point, if you remember from the previous slide. And then precipitation of the warmest quarter, the last variable that really seemed to distinguish these four, Louisianensis and the northern subspecies Meridionalis have pretty equivalent warmest quarter precipitation, but Calurdi prefers a much higher precipitation. So going on from there, second objective, actually developing these distribution models. Again, as to reiterate, I was using maximum entropy, this max-ent algorithm, and we presumed that these models would follow pretty well the bounds developed by Meacham in 1968, because he was the expert at the time, he had the most records at the time, so that's what he worked with. But also, my assessments, based off of everything that I had seen, brought me to think that Meridionalis, this northern subspecies, would be more impacted by soil characteristics than the southern subspecies. This has to do with the fact that reports have actually found them underground, especially when it's drier. And so if they're underground, that means that the soil characteristics are affecting them to a greater extent, or could affect them to a greater extent. So, distribution models, just as a brief overview, these, especially max-ent, it identifies areas of high probability of presence for the species. So it's not saying like this is a guarantee, this is where they are. It's saying that these are areas of high probability for them to exist in, based off of all of the variables that were brought in. Now, this requires occurrence point data and environmental data, and there are other algorithms that can be used for this distribution modeling, but these other algorithms have proved to be less robust with lower occurrence records, and considering how poorly understood the species is, I couldn't guarantee that I had enough records for these other models to be effective in. But more so, maximum entropy has shown to have a much greater ability to prevent overprediction, so saying like, it's 100 kilometers west when there's no way it's over there, it's on the other side of the mountain range, or something like that. So generalized linear model, domain, many others. I just stuck with So with this distribution modeling, again, went through all of the literature I could find, all of the museum records I could find, to make a list of every citation available on this species. Some of the problems that occurred, some of these locations are not well defined. We've got one of these up here that says 100 kilometers west of Tampico. You can't place that effectively. Some others, much better. 0.6 kilometers north of some cities. Like, that's great. I can work with that. But then others are mislabeled. And that's just to go to show how misunderstood this species is. I found records listing them in Virginia. I found records of them in Georgia. I found records from Florida. None of those are accurate. This species is specifically Texas and Mexico only. And so those records, again, highlighted in red because that's absolutely not the case for this species. Alright, so initial modeling on the my left, your right, I suppose. So these are the three different subspecies that I used as model to model. Now, in this shaded region here, actually, we've got the original range for the northern and southern subspecies, and I included Louisianensis in this analysis more as a comparison to see, like, you know, where does Louisianensis come down from? It's a species that's well understood, so we recognize their range, and also as a way to drive home the separation of Meridionalis as a species, so black-spotted newts, as its own species compared to viridescens. So in here you can see, like, more the darker colors represent a higher probability area, and then as it gets lighter, it's lower and lower probability. Now, I specifically wanted to refer to this other data that I collected during the course of this investigation, because I didn't use it in my modeling, but I did find it really intriguing at how well these ranges overlap. So if we move that over, you can really see how Clerty has a much higher prominence in this tropical forest ecoregion area, whereas Meridionalis, the northern subspecies, has a much greater prevalence in these Great Plains here in Texas and very northern Mexico. And so using that, kind of going back to what we discussed earlier with the environmental variables that are associated between these, you can kind of identify, like, if it's lower down there, it's in this forest, it's in this more tropical area, that would make sense for a much higher like rainfall over the course of the year. So now I want to focus on each subspecies specifically. This map is a much more refined representation of what the previous map showed. So this one is only showing between like 100% and 80% probability. The other one showed the whole range. So based off of this, you can see that there are roughly like 5 hotspots for the northern subspecies here in Texas. We've got right around Baffin Bay. We've got right along the coast in Willacy, Kennedy counties, and then over here in Hidalgo County, we've got another nice hotspot right about the border with Hidalgo and Cameron County, and then south central Cameron County, and then northern Tamaulipas states. These seem to be the hotspots in the records and also during this modeling procedure. Now for the southern subspecies, ran into a little bit more of an issue. So the northern species, I was able to use 32 records, pretty good. The southern subspecies, even less is known than the northern. So I was only able to find 17 citations that I felt comfortable including in this analysis. But even still, you can see there appear to be roughly 4 pretty strong and relatively isolated pockets of high probability for this subspecies. One dominant right along the coast, and that one coastal pocket seems to be pretty well separated. There's a little bridge there, but pretty well separated from the other 3 along the foothills of the Sierra Madres. And so, something, again, that I wanted to bring up with this is there was some slight over-prediction. So this area down here, this is Pico de Orizaba. Now, if you look on this from Google Maps or Google Earth, you'll see this is a pretty high mountain. It's likely a volcanic mountain. There are no known records from here, and personally I find it very hard to believe that they are actually there. But this just goes to show that this is a modeling algorithm. You can't be absolutely certain of every situation. But, on the other hand, this tiny little red zone here, this high probability pocket, does have a record that other individuals have referenced. Now, I do have that record, but I didn't feel comfortable in placing it. These individuals are actually from the area, so they have a much better understanding of where this might have been, and they placed it right where my model predicted high probability of them being. So that actually gives me some comfort in knowing that this model can generate really or fairly accurate representation of where they might be. Now, these models, Maxent likes to throw out a bunch of different numbers for you. Quickly here, we've got 4 different values going on with all of this. We've got a contribution factor, an importance factor, and then training gain values. So contribution and importance kind of go without saying. One is, this value or this variable is contributing X amount to the model itself, but it might not be so important, as you can see with the KCL mid-range. Relatively high contribution, about 16% of the contribution comes from that, but it is not important at all. But when you look at the training gain with only and compared to the training gain without, this is representative of like if the model was conducted without that variable, how much of it would actually, or how much would still be explained. And so you can see without that value, all of it's basically still explained. But on the other hand, training with only that value, it's still actually able to explain a fair amount for just a single variable. Now some of these, of course, are going to explain more. For meridianalis, elevation was a huge one. Even without elevation, the model was not able to predict nearly as well as it could have with elevation. And then for Calerti, had a few more variables that seem to have some importance to it. But again, much the same: contribution, importance, training gain values, and real quickly, didn't explain it before, but I ran two different models. So one is the constrictive model. Sounds a little counterintuitive, but it's including every variable in that modeling algorithm. So all the climate variables, all the soil variables, elevation, all of it. The generalized model, it's generally, well, the generalized model allows a slightly more expansive range because it's reducing the number of variables. So in order to generate this, ran correlation analyses, excluded all of the variables that correlated with already well documented variables from the previous analysis, and then based off of that, reran the model to get this, and you can still see that, in this case, warmest quarter still had a huge impact on the model, much like it did before. But now it's just slightly, a bit more emphasis on that one, particularly because some variables are correlated with it, so those correlative values are now going towards this, or towards that one. Lastly, for this part of my thesis, I wanted to look at the overlap zone. So there is a small zone of intergradation that was defined by Meacham in 1968. He defined this intergradation simply by two populations, where they appear to have morphometric characteristics more similar to the northern subspecies than the southern subspecies. Now, because it is much closer to the southern subspecies, I figured that it would be more likely that the southern subspecies would dominate in this intergradation zone. Oh, okay. So, what I did, within that intergradation zone, I took 1,000 random points, extracted the probability values for each of those, for each model, and then ran a t-test to compare between the two species, or between the two subspecies, rather. And so you can see, it doesn't really matter which model you run, t-test still comes out showing, yeah, it's almost certainly Calerti, just based off the probabilities. And if we were to go back to the actual distribution map that I generated, you'd see like this whole area is red, whereas up here, this was where all of meridianalis was. Now then, on to mitochondrial genetics, because let's do something completely different. So, for this first objective, we wanted to sequence and analyze the complete mitochondrial genome for each subspecies, and compare them to all of the other notothalamus species specifically. Because there have so far been 3 assessments of the entire notothalamus genus and the relations within it. The problem is, 2 of them disagree with the third one. Now this third one used strictly allozyme data, whereas the other 2 used very minimal mitochondrial genetics. And so we thought, we can get the full mitochondria, let's do that and run this analysis. So these are the 2 known phylogenies, again, we've got Nothothalamus genus all on its own, with Meridionalis being the most basal of the 3 species. Comparatively, though, when allozymes were analyzed in 1990, we saw Meridionalis sister groups with Perchriatus. This doesn't really make much sense from a geographic perspective because Perchriatus is specifically Florida and Meridionalis is Texas, whereas Viridescens is all the way in between those two. So you'd really think that from a geographic basis, Meridionalis should be more closely related to Viridescens unless it's the more basal group. And I'm going to get into that a bit later because some of that data is pretty exciting in my eyes. So here's a representation of what the mitochondrial genome looks like. We've got 22 different tRNA genes. Didn't show those because they're super tiny and it would just clog this whole thing up. 13 different protein coding regions here in black, and then the 12S, 16S rRNA coding regions up there, and the D-loop control region. Now, in order to sequence this, didn't want to just send the whole thing off. We wanted to make sure that we amplified the full thing so we had enough to work with. So, in order to do that, we divvied up the mitochondria into 3, I call them trits. So, you've got the L1C2, C2G, and then the GTOL. Now, these 3 are roughly between 5,500 to 6,000 base pairs. These PCRs took forever to run. And then once we got those finished, put them together, sent them off for next-gen sequencing, and aligned those with the GenBank with the GenBank accession for Meridionalis that was already constructed prior. So once we get the full sequence, we then have to partition that because each gene, each location of a codon within each gene is going to evolve at a different pace, because you're not necessarily going to have the first position of the codon mutate at the same rate as the third due to wobble and a lot of other factors. So using Partition Finder, ran the alignments, and we got this partitioning for the full mitochondria. You can see that a lot of the tRNAs had this GTR plus G method. Again, here GTR plus G, and then these few were HKY plus G. I'm not going to go too much into that because I've got more to go on. So, once we've got that partitioned and analyzed, we lined them all up and we assessed how close related via a percent similarity each sample that we had was, and across the species that we use as outgroups. So this is representative of 7 of the samples that we sent off. 2 of them came back as identical sequences. Not really, a little bit of an issue there, but what I want to point out here is Altamira is the only Mexico subspecies individual that we had. And when you compare that across all the other Texas ones, you're seeing a greater than 2% mitochondrial genetic difference across the whole thing. And that is pretty indicative of some decent separation between the 2 subspecies. Now within Texas, fairly little divergence actually, no more than 0.2% divergence within the individuals that we sequenced. But, as I had mentioned earlier, something about genetics that can come back and really intrigue you. This number here, so again, this is the Mexico subspecies. Compared to all of the other Texas subspecies individuals that we had, it actually has a closer similarity to the eastern newts than the Texas subspecies has to the eastern newts. So it's not by much, roughly by 0.2%, but as we've already seen, 0.2% is about how different all the Texas individuals are. So, got a little interesting for me there, trying to think of why that might have been. But, putting that aside, this is the final phylogeny that we came out with using full mitochondrial analysis. So you can see every Texas individual grouped together as one single clade with the Mexico individual as an outgroup for, or as a sister to that Meridionalis clade. And then from there, all of those clump together as one species clade and branches off from the other two notothalamus farther up, the viridescens and perstriatus. So, based on this, we can pretty conclusively say that yes, Meridionalis does seem to be sister to the other two species that are monophyletic. So, from there, went into try to identify, if we could see what the divergence date was between these two subspecies. Previous analysis by Weisrock et al., or not Weisrock, sorry, Whitmore et al. They went through and did a mitochondrial genetic analysis between specifically Iowa viridescens groups, because some glaciation events they thought may have separated them, and their analysis shows that they were pretty well separated by that glaciation. But they came out with a figure to estimate divergence state based off of percent identity. And based off of their analysis using our data, we were actually able to come up with an estimation for the subspecies, for the Meridionalis subspecies divergence state at around 1.8 to 1.5 million years ago. Now surveying. Again, something completely different. So, for surveying, as I had mentioned before, black-spotted newts are incredibly hard to find. Even when you know where to look for them, there is no guarantee that you're going to find them, particularly because they go underground, as I had mentioned before. So, thing is, when I first came down here, I was told I would not find a black-spotted newt unless there was water present. And so, to develop a surveying technique specifically geared towards identifying waterborne individuals, we developed an eDNA assay strategy in order to try and identify black-spotted newt presence. So in order to do this, just collect some water, run it through a filter, and then extract the DNA from that filter, run PCR on it. Pretty simple in concept. But there were a few snags. So, first we did some primer testing. We came out with 4 different primer sets, 2 of which are nested within one, so that allows for a greater amplification in case of very low yield from the first set. But from these 5, you can see, or from these 4, you can see primer set 5, 5.1a, 5.1b, and 7 all show bands at the appropriate size that we're expecting. So 5, we're expecting 276 base pairs. It's just below this 300 base pair line there. Primer sets 5.1a, 5.1b are around 186, 185 base pairs. Again, they're just below that 200 line, with primer set 7 at 171 base pairs, just beneath both of those, underneath that 200 base pair region. Now, extracting those just to be sure that our primers actually amplified what we thought they were, we sequenced those, and they came back as the correct sequences, so we were able to continue. That's where things kind of get a bit tricky. So at first we started running it with 4 different filter sizes. So by filter size, I'm specifically referring to the pore size of these filters. Typically for eDNA studies, they're using filters between 0.45 micrometer and 1.2 micrometer. Now we started with 1.2, used 3, 5, and 8 micrometer filters. The idea being that with a higher pore size, you're not likely to get as much DNA, but due to the water quality that is down here, you're likely going to clog some filters at those smaller pore sizes. But, as it turns out, all of those pores got clogged really quickly. So you can see here, 1.2 micrometer all the way down to 8 micrometer filled with less than 100 milliliters or less of water. Now, when you look at other eDNA studies that use like those 0.45 micrometer pore filters that I was talking about, they're running 2 liters of water through that. And it's like, oh, that's all great, nice for you. You're using clean glacial lake water or fresh flowing stream water. So you got like no particulate matter, nothing to block your pores with. That doesn't happen in this water. This water gets atrocious. So, as a way to try and get around that, we decided, what about a coffee filter? Coffee filters are roughly around 30 micron pores, or 30 micron in pore size, and they're cheap. Anyone can get them right off the shelf.
At Bed Bath & Beyond.
At Bed Bath & Beyond, apparently. So, we also tried running this water, this grimy muck water, through this coffee filter, and we were actually able to get around 1 liter, and in some cases more, water through that coffee filter. And when we run the extraction and run PCR on it, well, all of them come out, but the neat thing is still that coffee filters also came out quite strong, quite visible, and all of these sequence the appropriate region that we were targeting, which indicates that even the use of coffee filters, something that you can get like the hand press and about 100 filters for maybe about $10, you can perform the initial steps of eDNA. And that's incredible when you consider that those other fancy pores, or those other fancy filters, it's about $1 per filter. And so when you're trying to get 100 of those filters, the price really comes up, and you've got all of this all of these special items for trying to run it through. You've got this vacuum pump, you've got the container for where you're putting the filter on to try and suck it out, and you've also got to like carry around a battery in order to run the vacuum. Way too complicated. Much easier to just take this thing in hand with a few filters and walk out to the field site, because no guarantee you're going to get your car out there. Oh, that's weird. There we go. So, we've got an eDNA technique up and running. Second objective, try to find locations where black-spotted newts actually are. So again, going through historical records, literature, trying to really focus in on those sites. So this includes some sites like Laguna Atascosa, well-documented black-spotted newts, Sable Palm Sanctuary, Southmost Nature Conservancy, all of those well-documented in the literature for decades. Also, we're trying to do a comparison between trapping techniques, so we used cover boards, pitfall traps, borescope, which I'll get into in a little bit, and just general debris search surveys as the primary use that, or like the general herpetological surveying method. Continue on. So, these are all the sites that I surveyed and all the sites that I actually found things at. So these locations with field markers, that's where we were actually able to find black-spotted newts or get samples from. And then everything else, all of those we surveyed, we didn't find anything. If it's circular, it's indicative that there is a historical record for that site. If it's square, there was no historical record at that location before. And this is actually an old picture from Rapol and Klicka surveying Laguna Atascosa for black-spotted newts. They actually estimated roughly 500 larvae in that pond. I haven't been able to find any newts at that pond during my time, but they were able to get one before I arrived. So, during these surveys, again, as I mentioned, visual encounter, debris search, coverboard surveys, pitfall traps, borescopes. When we collect an individual, take a tissue sample for the genetic analysis, weigh them, measure the length, or SBL, so snout to vent, specifically right up to right where their legs are, just about. And also took a ventral photo for future recollection identification. Every individual has a unique spotting pattern, and I'm actually able to go through and identify if we've recaught an individual at a certain location based off of those markings. Then, borescope surveys. So, borescopes are generally used for like wall maintenance or automechanical repairs. But what's handy about them is they fit into these very tight, small spaces, which is needed when you've got something that's going underground into a crack or a burrow. Now, because they keep going underground, that's generally the reason why they're so difficult to find. You can't just go walking around and naturally flip something and find them if there's a crack nearby. They're going to be in that crack. So I wanted some way of trying to look inside of that crack to try and find them. And so I thought, why not use this borescope? It's about $100, takes photo, video, and we were actually able to find newts down in these cracks. This one's kind of running away from me here. But it's actually fairly effective. You're able to find black-spotted newts using this borescope. Just takes a little effort to get used to it. So, our survey results from the samples that I personally surveyed and actually had newts at them. We've got 2 sites that had huge numbers. Specifically, the Sal del Rey site, that's not its actual location, that's private information, and then Palo Alto National Battlefield. These 2 sites, great number of newts. It's fantastic. However, something you'll notice, these 3 historical sites weren't able to find many newts at. Now, that isn't necessarily because they aren't there. We know they are there. We found them. It's just that some of these sites, it's a little bit more difficult to survey for them. Because, especially at Sable Palm and Southmost, it's dense forest vegetation. Where are you going to go to try and survey for these things? You've got leaf litter everywhere. You've got dense, thick shrubbery everywhere, so you can't get around as easily. And we don't really have a specific location of which water body they're using to breed in, so we don't know where to focus our searches. Just a few minor details.
Okay.
Yeah. Something else to note about all of these, 4 of these sites that we found them in are generally like low depression, ephemeral ponds, or Resaca beds. And that actually I found kind of interesting, especially considering Cameron County is full of these old Resaca systems. Go down. Oh my gosh. Can I just close it? I'm just going to close it. Okay. So, technique comparison now. So, only 4 of these different techniques actually managed to find anything. That's the cover bore surveys, the debris searches, the borescope, and visual surveys. And those visual surveys were actually kind of a fluke. Because we were finding newts under debris and then just happened to look up and there were 2 more newts popping out of a burrow. So that was kind of lucky. But something that we found in this is that tires and this woody debris really seem to give the most newts for what you're looking for. And these tires, they're just random debris tires. People just toss them on the side of the road. And that's where the newts were. Cover boards in total produced about 12 newts. Borescope surveys produced, I believe it was 9 newts? Yeah, 9 newts. One of which was actually in an artificial burrow that I constructed as a way to try to supplement the borescope surveys, because we know that they were using these burrows, so why not try to make burrows and use that as more of a passive trap using the borescope to identify them. 7, sorry. So, another fun thing about the borescopes is that you're actually able to find a whole ecology underground. So, while I was able to find black-spotted newts, I was also able to find toads, snails, slugs, ants, specifically invasive fire ants. Not so great, but it's good to know they're there. Rats in some of these cracks, like these are big fissures, like yay big some of them. you could fall in and break your ankle. But rodents seem to like them, centipedes, millipedes, even a few spiders. There are a few tarantulas that I've actually come across that were quite perturbed at this bright light shining on them. And so that's actually pretty interesting to find that there's this whole ecology underground that you're able to survey now with this borescope technology. And it also can give you an indication of what some of their what some prey items might be and what some predators might be. So we actually know that black-spotted newts feed on snails. One of the first individuals that we found pooped out a snail shell for us. So we're positive they eat snails. So when we find a snail down in there, it's just like, hey, there we go, that's food. Centipedes, millipedes, potential prey. We found pillbugs, also potential prey items. But some of these spiders, especially tarantula, maybe that's a predator for them. We found a snake down in there, also a potential predator. So we get a bit better of a sense of what the ecology is like when we can't even see them. So, as an added bonus, because we were taking these weight and length measurements, why not just do a weight-length trend? Because that's a pretty standard thing to do, and when you don't know anything about a species, anything new is positive. So, doing this trend line, you can see that there are a lot of individuals that really weigh between 2 and 4 grams and are roughly between 4 and 6 centimeters in SBL length. These two images are actually the largest and smallest individual that I ever came across during these surveys. And you can see the scale here is trying to represent the actual two-scale comparison between these two. This one's roughly the size of her entire, just her body, not even including the tail. And so this individual actually, I believe, is the largest black-spotted newts on record as well. Something I did find a little interesting, just as a side note, is that we know black-spotted newts can live to roughly 20 years, but we know that through aquarium individuals, and those individuals were not this size. So it's not necessarily an age to weight or length ratio. Then some of the habitats that we found them in, because again, more information is best. So we managed to find black-spotted newts in these ephemeral ponds, sable palm forests, these Rasaka beds, all wonderful. But then, something a bit more interesting, we're finding them in backyard ponds, a little bit more manicured, a little bit less like what you would expect. But then, more specifically, in this agriculture field. That agriculture field is one of my best sites for black-spotted newts. It's been that way for 30 years, and yet they're still hanging around. This site, because of this low-lying depression, after heavy rains, it will fill up, make a water body there that they just can't plow, they can't plant in, and so it's just enough for black-spotted newts to actually use and breed in, or seemingly breed in. I mean, they're still active there. So some of the concerns that I came across when doing these surveys. Coverboards, it's great, they are effective, but because of their size, they seem to actually prevent moisture from seeping into the ground directly underneath. Now, in one sense, this is providing microhabitat refuge for the newts, but even with the borescope, it's still hard to find them down there, especially as it gets deeper and deeper. So these larger coverboards, effective, but also cause these cracks, which makes it much more difficult to actually find them. Pitfall traps were completely ineffective. Now, previous studies had actually indicated this. Rapul and Klicka tried to use coverboards, or excuse me, tried to use pitfall traps, but they were finding newts crawling underneath the fencing or underneath the buckets instead of falling into the buckets. So that's not really going to work out for us. Also, we don't really know how climate change is going to impact them. As I mentioned prior, these coverboards really preventing the moisture, forming these huge fissures, but without understanding the effect of climate change on South Texas, we don't know how dry it's going to be, how wet it's going to be, so we can't make a valuable assessment on how black-spotted newts are going to be impacted. And lastly, this presence of invasive fire ants is a bit concerning. Fire ants are known to be really aggressive and will basically eat anything that they manage to kill when they come across it. And considering that they're found underground in the same locations that newts are found underground, that could cause total chaos for any newt that runs across them. But as a bit more promising, some of the conservation implications regarding this is that, again, we were able to find newts in many of these artificial or really impacted terrains. So specifically here, you see this water body. Newts are known from this water body, but it's a cattle dugout, and it's within this old Resaca system bed. So what we can ascertain from that is, you know, if you're gonna try and make a cattle pond, maybe make it in this low-lying depression or this Resaca bed, because we already know that newts seem to prefer these systems. This image in particular I wanted to point out because there are actually 3 known sites within this one system, and they're all in this Resaca bed. All of them. Like, I've actually surveyed in other sites that are outside of that Resaca bed, but I was not able to find any newts at this location where we know there are newts. And then soil salinity effects, relatively unknown. This site in particular has relatively high saline soils. Like, the only thing that grows in those soils are super soil, or incredibly salt-tolerant plants, and yet newts are in that soil. So they might be able to deal with it in one fashion or another. But also, considering that I was actually able to discover 3 new sites with black-spotted newts, in addition to continued surveys of historical sites, it might be that newts are a bit more prevalent than we think. It's just that they're hard to find. They go underground, you can't really see them, and you need the absolute best conditions to get them. So we might be underestimating their actual area, or their actual populations. So from this, I mean, we found that the distribution models generated were able to fairly well predict where black-spotted newts are, especially in that Clarity example, where we didn't have any record for it, but it still predicted that one site. The intergrade zone seems to have a much higher propensity for the Mexican subspecies, which we'll have to do some genetics on those if we can get them to really clarify that relationship in that zone. Genetics assessments across the whole range really shows that, yes, black-spotted newts are the basal species to the other notothalamus species, and the two subspecies are pretty well distinct from a genetic basis. Along with genetics, we've got the environmental data, which similarly shows that there are environmental factors that affect the northern subspecies more than the southern subspecies, and vice versa. And they also have different preferences for some environmental variables, either higher in one compared to lower in another. Same, all that. And then with the addition of the eDNA assays and the borescope surveys that I presented here, I believe that we will be able to more readily be able to, or we will more readily be able to detect presence or absence of, well, presence of black-spotted newts in certain water bodies. So in order to identify just how far-ranging they are, what kind of water body systems they actually are able to be maintained in, and from there go on to say, water quality samples, habitat assessments, all of those important things. So, future directions, again, collecting more samples. More samples, always best, get more of that data. Continued monitoring of the populations that have been identified, because, again, so little is known. We don't actually understand their population sizes. We don't understand their life history cycles very well. intragenetic comparison still needed, and especially give some focus to these agricultural sites, because if they're able to be maintained in these agricultural sites, there's a bit more hope than was previously thought. Subspecies clarification, mostly just we managed to use 3 individuals from the Mexican subspecies, but there are what appear to be 2 distinct ranges for the Mexican subspecies. So greater sampling in that would be required to get a much greater assessment of their genetic relations. And yeah, life history. We don't know much about it. We know that they go from egg, larvae to adult, and then underground. And then aside from that, that's about it, really. So, I want to thank you all for listening. Thank you all to all of the different park managers that helped me out with this. Thank you to all of the GPZ crew that helped me do all of this work, and all of the assistants, because I know that some of you have come out with me on multiple occasions and managed to find newts on your own or with me. Either way, all of the help is appreciated. And so with that, I'll take any questions.
Why did it look like in some of the bodies were included like that? Like on that last slide?
That, this is actually the
Hey,
only...
repeat the question so that...
Oh, yeah. So the question is, why are some of the... or why do we have some images of individuals in this contorted fashion? Some of that is just when you're handling them, they're going to be all floppy. They don't really stand still very easily. But this individual on its own actually contorted into this fashion, showing that bright coloration of its tail. Now, in other newt species, especially the Tarica, the related genera in the West Coast, they will do this sort of flexion, but more so like chin up, tail up, to accentuate that bright body color. And that's an indication of like, hey, I'm toxic. This is an aposematic coloration scheme. The eastern newts, pretty well known for being toxic. Tarica, really highly toxic. Multiple accounts of individuals dying from slight ingestion of the toxin, people going blind from rubbing it in their eyes. Basically, handle them, just wash your hands before doing anything. That's really all we ask. And so this individual is kind of doing that same sort of fashion, showing off that bright tail. There have been, Rapol and Klicka documented a very slight neurotoxic capability when rubbing them against their lips. Their lips went numb. And so maybe some future studies involved in that, but I think the focus for now should be more on the conservation and life history aspects revolving around this species.
You mentioned earlier that the Mexican subspecies is more related to the eastern.
Yes, it appears through genetic, based off the genetic data that we have, it does look like there is that the Mexican subspecies is slightly more related to the eastern newts than the Texas subspecies is related to eastern newts, and you really would not expect that. Now, I have my own hypotheses regarding that. In my mind, I believe that, especially considering that the previous study that did the divergence date analysis back here, once I get to it, anyways, they estimated the divergence between the two genera to be roughly 11.4 million years ago. Yeah, here. So you can see here on the scale N. meridionalis, and then this is Notophthalmus viridescens as a whole, including 2 of the different subspecies for viridescens. And right around here is their estimated branch off at They say 11.8 million years. There doesn't quite fit with this bar. Some distortion might have occurred when I copied the image over. But based off of how old that separation was and the apparent like genetic similarities that we actually found between Mexican and Eastern newts, my hypothesis is that it all used to be, one contiguous range, but then due to a glaciation event that cut off the Mexican branch of that clade, they all got pushed down into Mexico. After all, black-spotted newts cannot handle the cold. Previous study tried to do, it was very specific. It was sexual dimorphisms in response to temperature gradients across 4 different gradient schemes. And in one of them, it was the cold gradient scheme, where they went down to 5 Celsius, and all of their black-spotted newts died. Just all of them. And again, we're finding eastern newts under the ice, and just swimming around fine. So, based on that, it would seem like they likely got pushed down due to this glaciation event, continued evolving down there, and then as the glaciation went away, and more recent times, they've been able to migrate back up north, and then nearly overlap with what is present-day eastern newt range.
Question and a comment from Dr. Busenkamp. First, he says, all models are wrong. Some models are useful. How do you respond to that?
Fair point. As we saw in Clarity, there was some points of overprediction. So it's not that it's perfectly accurate. It's trying to provide a assessment of where they are more prevalent, not necessarily this is the absolute range that they are found in. So it's useful for trying to find this is supposedly a hotspot. We need to focus our surveys there rather than we have this entire range to survey and we have to provide equal surveying focus to all of it. It's if you have an idea of where a higher probability is, maybe focus on that first.
So some ground truthing is in order for this type of thing.
Oh yeah.
And also, he says, so since you've got now, looks like 2 subspecies, should they be managed as 1 species or 2?
Oh man, that.
So if we wipe them all out of Texas, is that okay? Do you still have some down there?
Well, we'll focus on Texas for now. So within specifically southern Texas, there are 3 salamander species. There's black-spotted newt, there's the Texas siren, and there's the tiger salamander, or the barred tiger salamander specifically. Each one of these 3 species has its own particular niche. So sirens, they handle the desiccation of southern Texas by cocooning themselves underground. They're excellent for identifying where water bodies might be. Black-spotted newts, based off of prior estimates, are really good indicators for what the water quality might be. And then the barred tiger salamander doesn't actually come. The range claims it comes down into Cameron County. I have never found them in Cameron County. I don't know of many records of them in Cameron County, but needless to say, they exhibit similarly. They exhibit different ecological niches primarily. So, tiger salamanders are able to feed. In theory, would be able to feed on black-spotted newts, and sirens generally feed on anything that they can come across. They're pretty omnivorous in that regard. But as far as managing them separately, that's a tough one because they are definitely distinct subspecies, but as a whole. I think, yeah, it comes down to they each have their own specific ecological niche. As mentioned before, the southern subspecies predominates in these tropical forest regions, whereas black-spotted newts are far more dominant in these Great Plains regions. And so by managing them, by recognizing that they both need to be managed distinctly or separately, you're also recognizing that these two ecoregions are distinct ecoregions. You wouldn't manage forests and grasslands in the same manner. So by that same assessment, you shouldn't manage these two subspecies that are separated by these ecoregions in the same manner. I hope that works for them. Of course you would ask that sort of question. Yes?
So, can you expand on the soil parameters you said you used for
Yes.
your taxon comparison, and which were those?
So for soil parameters, it came down to I listed them as two different classes of soil parameters. There was the chemistry soil parameters, or chemical, and then particulate matter. So for chemical, we had pH levels, specifically either measured through H2O pH or potassium chloride pH, which I wasn't aware was a thing, but it was available, so it's like, I'm going to include that too because I've got this data now. And then soil particulate matter, so we've got bulk density, we've got coarse fragmentation, we've got sand, clay, silt parameters, and actually there was also an organic carbon content parameter. And each one of those soil parameters was also assessed at 3 different levels: at surface, at 30 centimeters below ground, and then at 1 meter below ground. And this was done because we understood that black-spotted newts use these underground fissures for their microhabitat or for refuge. So by trying to assess by assessing the soil parameters at those deeper states, maybe would have come up with something. It didn't really seem to, though. There's a lot of correlation between those.
And salinity, soil salinity was not one of those parameters?
I didn't have the source that I used for these soil parameters did not provide a specific soil salinity measurements. I do believe that soil salinity has a major role to play in black-spotted newt habitation, but there are some interesting caveats in that soil salinity plays a role insofar as I believe it increases the rate of salinization of the water body itself. Because what we were able to find was, again, at Palo Alto, very saline soils there, and yet there are still plenty of newts under that ground. So they seem to be able to handle relatively high levels of salinity. However, Rapol and Klicka, when they did their assessment, they found that black-spotted newts were not present in water bodies with a soil salinity higher than, I believe it was 1,000 parts per trillion. And so in these higher salinity soils, my understanding is that there's going to be a freshwater, saltwater barrier that comes into play after the initial heavy rains come down. There's this fresh water on top. That's when the newts go in, breed, and then leave. But as that water drains out due to evaporation or just slowly over time, the salt will percolate into that water body, making it brackish and then making it more saline to the point where they might not be able to handle it specifically in the water.
Did you measure soil salinity in those locations and in those moments where you found newts?
No, we did not have a salinity measure apparatus at that time.
Take a sample.
Yeah, no, that's something that we're definitely planning on doing.
But it would be interesting because that would answer some of those questions. So to say salinity may be affecting these species.
Oh yeah,
And it's
absolutely.
exactly where you found them, in the moment where you found them, which may change, but sometimes they are bigger.
Yeah.
If you take a sample of that location where you found them, you got to answer that question.
Mhm.
So should we just throw tires out everywhere and make more newts?
Well, so the tires at that one site were kind of a lucky happenstance for us. No, I do not suggest just tossing tires out. there's definite concerns for whatever chemicals might leach out of those tires. But something that I do want to, since we're on the topic now, bring up regarding those tires in comparison to the cover boards. Tires and these branches and the bark that we found newts under, they all seem to have a relatively thin footprint. So I'm talking like, you know, roughly around 20 centimeters or less of a footprint. This is a small enough footprint to allow the rainwater to actually percolate in, like, farther in to the ground. So like that's directly underneath whatever cover it is, while not protecting a huge portion, or like while not protecting too much that would then later fissure underneath. And so my suggestion is either use cover boards with like holes in them or perforations to allow a greater amount of water and rain to get underneath those boards, or I mean just make long thin boards or tire or like rounded boards, because that way the water can actually seep underneath and moisten that up. And part of the reason why I think those tires really helped is because that moisture was able to get underneath, it actually drove the newts up and onto the surface. And that's why they were so much easier to find.
Do you think, there wasn't porosity data available for the maps, correct? For soil
Like how
porosity?
refined it is?
Like the spacing between individual sediment grains. So, the reason I guess I ask is because I wonder if that might influence the distribution of where you're finding them as well. Because it might...
Oh, okay, so like, so you're asking about like, if there's measurements of like the percentage of each different...
Just how much pore spaces there are in between the granules. So it's I know that sometimes...
Oh, okay.
Because I wonder if that would influence whether or not it's easier for them to burrow.
Yeah, so question regarding...
That's more involved.
Yeah, so question regarding the porosity of the soil at these locations. That data wasn't really present, but with an individual that might have a much better understanding of soil mechanics, you might be able to assess that, because each of the like sand, silt, clay, coarse material, all of those came as percentages of total soil like prevalence. And so, you know, you add all of those up, it's going to be 100%. But some of these areas, it's much higher sand percentage, much higher clay percentage. Some of them also, you know, it all varies to that extent. So I don't have the understanding to make those assessments, make those judgments, and they weren't provided for me. So I just went off of the percentage available at all those sites.
The other thing I was going to ask, so I remember when you were first looking for these things, trying to figure out what they were, there's a mention of crawfish burrows that they would inhabit, right?
Yes.
Did you find that when you found these subterraneously, did most of them occur in those fissures that can drive more so than other?
Most occurred in the fissures. The ones that I found mostly occurred in the fissures. I want to be specific about that because we did find individuals in burrows. We found two during our visual surveys crawling out of a burrow. We also found two more crawling out of a burrow that was directly underneath a cover board. And then there was one individual that I did also record deeper into a burrow. Now the problem is that those crawfish burrows and fiddler crab burrows, they're much more winding, twisty, and muddy. So once you get the borescope in there, it's pretty difficult to actually maneuver it around corners because it's a stiff neck. These fissures, they're straight down, they're deep, and you can avoid those edges so as not to obscure the view of the borescope. So these fissures are easier, in my assessment, to survey using this borescope, but all the same, the fact that we know that they're down there and we have a way to actually survey for them down there, it's still something to take into consideration.
And considering what you know, do you think that they might have a preference? Or not enough, maybe not enough.
Not enough data for that. I don't think that you would find much of a preference necessarily. I think it has more to do with where the moisture barrier is, because these cracks can get really deep. I've stuck my entire arm into a crack and I couldn't feel the bottom. But because these cracks are also much larger, they're more prone to desiccation deeper down. So you might find that a newt is, like, on the same day, same location, my guess is that you would find a newt higher up in the soil column in a burrow simply because it's a much tighter space, and because it's very windy, twisty, it's much less apt to desiccation than these straight down large fissures in the soil.
There are no more questions. I'll move on to our next portion of the exam.
This is where I get reamed.
Thank you guys for coming.